Good evening and welcome. I want to make sure to extend a warm welcome to all from the KAUST community that are here, but we also have a large number of visitors from outside of KAUST. If the visitors all just put your hands up and so the KAUST people can know who to say hi to and welcome. So let's welcome all of our visitors from outside of KAUST. This is the final keynote talk and the final event of the Enrichment in the Fall program. So it's important to have a very special thank you to Professor Omar Canillo for all his efforts to help organize this, for Mari Lohr and the entire Enrichment Programs office, and also for one of our VPs, Yves Nanu, who's been very supportive of all this as well. So let's give all of those people a big round of applause. Thank you for a wonderful program. There's one other group that I wanted to be sure not to forget to thank. There have been several visitors and FEP events, and there have been a group of hosts that have helped to look after these visitors. So I wanted to be sure to acknowledge them. So starting with Shannon Brown, who's been hosting Gustav, Tane Sinclair Taylor, Royal Hardenstein, Allison Monroe, Tulia Terranillo, Morgan Bennett Smith, and Sofia Mudrova. Thanks to all of you for helping with the special visitors that we've had. And again, another round of applause for all the FEP speakers, the enrichment speakers that we've had over the last couple of days. Has been great. So thanks very much. And moving along to introduce tonight's speaker, it's a very special honor to introduce Professor Gustav Paulay. Gustav is a Yale graduate and then went to the University of Washington to do his PhD. And if you think I'm reading the wrong set of notes, if you were paying attention earlier today, that's the same path that Kathy McFadden had. So they both went to Yale. They both then went to the University of Washington for their PhD, and they overlapped at Washington as graduate students. And in fact, I understand that Gustav was Kathy's TA in an invert biology class. So there's, and I did not know that until they were here and told me that just a couple of days ago. So I think that's a great coincidence. After his PhD, Gustav went to the Smithsonian for a postdoc, and then went to the University of Guam, in a really cool place where he was based for 9 years. And then he moved to Florida and has been there since 2000. So he's been 18 years in Florida. He is the Curator of Invertebrates at the Florida Museum of Natural History, which is associated with the University of Florida, where he is also a professor. He studies the diversity and diversification of marine life. He is a regular participant in huge collaborations all over the world with really large teams who try to document as much biodiversity as possible. And in that process, he has collected tens of thousands of species, especially from coral reef habitats. He is a walking encyclopedia of invertebrate knowledge. It's really phenomenal. And I think we're going to hear some cool stories about some cool invertebrates tonight. In addition to having done work in the Red Sea, he's been all over the place, but just a selection of the exciting places he's worked have been in Oman, Australia, the Philippines. He's been in all Indonesias, Melanesia, Micronesia, Polynesia, including Hawaii. He's sampled throughout the Caribbean, the Bahamas, South America, and of course, both coasts of the United States. His aim through all of this work is to try to make biodiversity more accessible to diverse audiences and in diverse applications. Overall, his work is super important. It really broadly helps all of us understand how diversity and distributions of marine life came to be. So it's really special to have Gustav back in KAUST, and let's give him a warm welcome. We look forward to your keynote. Thanks, Gustav.
Thank you very much, Mike, and thank you for everybody for coming to this talk. And it's an incredible honor to stand up here and talk to you guys about reef diversity. It's such an amazing topic to have a the Fall Enrichment Program devoted to reef diversity, so I couldn't be more happy than to give a talk like this. So as Mike said, I'm really interested in diversity. There's no organism that leaves me cold. I like everything. And of course, if you're interested in diversity, a lot of that diversity is in marine environments, on coral reefs, the most diverse habitats, and it's in this group that some people think of as a group called invertebrates, which is basically just animals beyond fish, right? So what I want to talk to you about is what is on reefs, what kind of organisms live there, how do they make a living, where are they, and so forth. And of course, the first question you want to ask is, why would somebody study biodiversity? Well, first of all, it's really interesting and cool. You meet a lot of very interesting characters. But more importantly for us humans, biodiversity is what makes the planet tick. It's what makes ecosystems function. There's an incredible link between ecosystem function and biodiversity. Measuring biodiversity tells you how well you're doing with your ecosystem, and of course, you want to ensure it's there. Today, with the world changing as fast as it is, it's also really urgent because we're going to lose a lot of biodiversity. Ecosystems are going to be altered by the coming onslaught of all the change that we as humans have brought on this planet. And so today is kind of the time to look at the dodos that will not be here tomorrow. And also to gain that knowledge so future generations can manage the biosphere as well as possible. So that's kind of what's driving all of this, okay? So I would like to start by just telling you a little bit about what we know today. So marine biodiversity, there are about 250,000 or 250,000 described marine species. This is how many species scientists have named in the world. And out of all of that, 3/4 are animals that lack a backbone, which we call invertebrates. Diversity is inversely related to latitude, and it's highest in the Pacific, which is this area from the Red Sea out into the Central Pacific Ocean. And if you learn your fishes in the Red Sea, you kind of can identify a fair number of fish in Hawaii, because it's that well connected at some level. And finally, diversity is highest on coral reefs. So how diverse is the ocean beyond this, what we know today? Well, the estimated number of species, Kathy has just talked about this, so I don't even have to show this slide, is in the millions. But we don't really know how many there are. But what we know is that we know only a fraction of what is out there for a lot of different reasons. So it's really important that we get to know more of these players that make ecosystems what they are. And on coral reefs is where the diversity is highest. When you look at a coral reef, most divers that go on a reef see this. It's incredible. I mean, you've seen plenty of slides in the last couple of days like this. What strikes you is incredible abundance of fish and corals. And very large portion of coral reef science, management, thinking, recreation is focused on corals and fish. If you look at the science, people measure corals and fish. And that's just, what is out there, that's what you see, and that's what you study. But corals and fish are just a fraction of what is really in the world. This is the 250,000 species that are described today fall out roughly like this. About half of the species are mollusks and arthropods. And then vertebrates and cnidarians, to which corals belong, are just little bits on that. In the Red Sea, there are about 350 coral species, currently documented about 1,100 reef fish, and 6,300 other taxa. And while the corals and the fishes are relatively well known, although there's still plenty of novelty there, the other taxa are not. So let's look at these numbers again. On a reef level, there are less than 1,000 coral species in the world's reefs, talking about stony corals in this case. Kathy used that term a little more broadly for soft corals as well. There's less than 10,000 species of reef fishes, and then there's probably about 1 million other animals on reefs. So we're missing 99% when you look at corals and fish. So what I want to do today is tell you about the other 99%. Where are they? Who are they? And why are they where they are? Okay? Basically, another way to put it is, what do all those corals and fish eat? And if you want to see the rest of the diversity on a reef, you have to go inside it. You have to go into the matrix. If you look at the reef, it's an accumulation of skeleton. There's just a ton of calcium carbonate. Kathy was describing how corals accumulate the reef itself. Reefs can be miles thick, and both biological and chemical processes make pores in the reef, and it's like a giant piece of Swiss cheese, and it's completely permeated by living organisms. Once you enter the reef, through these little crevices, and in some cases in caves, humans can actually enter it. Otherwise, you have to study it in a more invasive manner. You see really a very different world, an alternate reality of what makes up a reef. And this is a very typical picture of the inside of a reef. What you're looking at, all these colorful colonies are sea squirts, there are sponges, there are protists, there are all sorts of animals that most people have not seen or even recognized as animals. And really, this is what the reef is about. It's about this deep diversity of animal life. And I want to just introduce animal diversity a little bit so what comes will make more sense to you. On the left is a genealogy, a tree of life, if you will, of the animal diversity on the planet. There are about 30, 34 big groups of animals, the biggest units of animal diversity that people recognize. They're called phyla. They include things that are mollusks. Mollusks include clams, snails, squid. They include things like arthropods, everything from spiders to crabs to copepods to barnacles. They include things like cnidarians, which include jellyfish and corals and hydroids. On this graph, if you look somewhere up on top in green, 3 units together are separates, cephalochordates, tunicans, and craniates. Those are the chordates, and in most books, those are looked at as a single phylum, and the craniates are what contain the vertebrates. Okay, so everything else is an invertebrate. So let's look at some of these organisms, so just to get a feeling for animal diversity. So sponges are one of the earliest branches of animal life, and these are animals that look like plants. They were thought to be plants when they were first described. They have no guts, they have no mouths, they have no muscles, no nerves, and yet they're animals and they live on the reef. So I have a little movie here from The Shape of Life by David Attenborough that gives you an appreciation of how sponges make a living. They're filter feeders. Their bodies are covered with little pores. They draw water through their body with little beading cilia and filter out the water that comes through them.
To make the invisible visible, Diaz injects a harmless colored dye into the water near the body of a sponge.
One of the ways we can test for the rate of water flow that moves through the sponge body is to inject a colored dye and measure the speed at which it is pumped through the sponge. When I start seeing the colored dye coming out of the sponge in less than 2 seconds from when it was applied, I could not believe my eyes. This steady and strong continuous flow of water continuously coming out of the sponge was an incredible realization of the dynamic existence of this organism. It's an incredible sight to witness.
They're gigantic filter feeders. They sponge out all the plankton.
These ghostly exhalations are proof that the sponge is actively pumping. With no mouth, it sucks water directly through its sponge-like body walls, which are shot through with millions of narrow canals and tiny chambers.
Okay, so I will move on to some other animals that live sessile, attached to the reef. You heard about corals and sponges, but there's a whole bunch of animal groups that live literally attached to the reef. One of them are the sea squirts. And sea squirts are our cousin. They share the same phylum that we belong in, the Chordata. And they changed so much because they stole cellulose from bacteria. They actually stole the cellular synthase gene from bacteria, incorporated in their body, and it allows them to make this cellulose-based external covering. And they're effectively these giant gill baskets. You can think about them as just a giant gill chamber with the guts attached from a fish. And that's what they really are. You can imagine an anchovy and throw away the rest of it except for the gill chamber and the guts, and you got a a sea squirt left over it. And I found this quote in one of the websites that I was looking for good pictures. Meet the creature that eats its own brain. And that's basically because they start out as a larva that has a brain, or at least a dorsal nerve cord and a tail, and swims around, attaches to the bottom, and then throws away all of that part of the body, all the muscles, their backbone, and everything else, and becomes this filter-feeding lump. And if you look at the recent phylogenetic results, the ascidians, which are colored in blue, are our first cousins. They're even more closely related to vertebrates than lancelets are, which are very fish-like. And there's a picture of a lancelet up on the top of the figure. Some of the sea squirts then become colonial, just like corals do. There's a whole bunch of different individuals that live together, and you can imagine them as a whole bunch of herring guts in jello, right? They take the cellulose covering and they embedded their guts in it into a colony, and each of these little pores on this diagram is the mouth of one organism, and then they all filter water, and the exhalant current comes out through the larger holes in some cases, or so forth. There's a diagram of a ascidian colony on the upper right that shows the basic architecture of these colonies. Other interesting colonial organisms include things like bryozoans, moss animals. Most of you probably have never heard of these. An interesting aspect of some of these sessile colonial animals is that they can specialize the zooids that are connected together for different functions. In this case, some of them become little bird head beaky things called avicularia, which are specialized zooids that have lost the tentacles and just fight things that try to land on the coral. They're basically defensive zooids. Others become brood chambers and hold the babies, and then you have the regular feeding zooids with tentacles that feed. So you can have incredible changes in the body when you're a sessile organism and do things in very different ways. Worms. If you're in invertebrate zoology, you're into worms. Here's the animal phylogeny I showed you before with the 30-odd animal phyla. Every one with a star against it is either all worms or includes some worms. So when you hear worm, you're basically talking about the basic architecture of an animal. And here is, for example, 6 different phyla of worms. They all look alike, maybe, at first, but they're very, very different organisms. One in the upper left is more closely related to you than to any of the other worms below it. Worms include things that are segmented, like segmented worms. There's a fireworm from a reef. These have really sharp calcareous bristles that are loaded with venom, which is why they are called fireworms. You touch them, and you get a pretty good sting from them. They include scale worms that have incredible scales all through their body and are carnivorous, and they can pop out the interior of their gut as a reversible pharynx with soft, fleshy teeth on the end and swallow hapless other worms or even larger organisms that come by. Some of these worms will even feed on fishes. There are flatworms that are totally flattened and incredibly colorful. This group of flatworms I'm showing you here feed on sea squirts. They pick up toxins from the sea squirts, and they advertise that toxin through their colorfulness. They can also swim through the water. The bottom left picture shows a swimming flatworm. They swim like flying carpets. So we have living flying carpets out there in the case of these animals. There are ribbon worms. These are rowing predators that have another proboscis that can shut out of their body that's longer than the body itself, and then they wrap around and envenomate their prey. They grow big. Here is one we got in the Caribbean. It was 4.5 meters long, and that's a little one. These things are known to be over 30 meters long, okay? And there's one they're very contractile that's curled up. Actually, that's the same specimen that we are holding in this picture. Brittle stars. There's an incredible abundance and diversity of brittle stars on reefs. And you hardly ever see them because they're in the middle of the reef. People have done experiments on these, and if you put them out on the reef surface on a tether, they're gone within seconds. The fish just gobble them up. We do a lot of samples of coral reef matrices, where we'll take apart a reef and look inside a half by half meter area. And from a half by half meter area, this is how many brittle stars we got out. Okay, so this is what's inside the reef. And there's not only abundance, but a tremendous amount of diversity represented in the symbol. Then there are the pods. All the things, pod means leg. There are amphipods, there are copepods, there are There are isopods, there are stomatopods, an incredible abundance of arthropods on reefs. And this is just a sampling of that diversity. Crabs, oh my gosh. When I lived in Guam, I tried to document the marine life there, and I have enough time to get into crabs a little bit. By the time I left, we had 400 species of crabs documented on the island. Copepods, oh my God. So this is where diversity is on a reef. And actually, there's some really exciting research right now being done at CAST by Sophia, who's studying copepods that live on living corals. And she has already got 200 species just from a couple of coral species on the reef. These are tiny animals. And if you think these are, well, okay, it's just bizarre, who cares? David Bellwood's lab has done some interesting work to look at how many of these pods are in a square meter of reef. Okay, so they enumerated what they found in there. The harpectocoids are copepods. Look at that, 300,000 copepods per square meter of reef. That's what's in the matrix. Ostrocods, Tenaes, these are all crustacean groups. Just incredible numbers. This is what the whole system runs on. These invertebrates are what feed the ecosystem. If you go into the inside of the reef, you also found things that are effectively living fossils. On the left-hand side is a coralline sponge, a sponge that actually lays down a hard calcareous skeleton just like a coral. In the Mesozoic, in the age of dinosaurs, these things formed reefs. Okay? Today, they are only found inside the reef matrix. On the upper right are brachiopods, same kind of story. Their heydays was in the Paleozoic. On the bottom is a phylocarid, which is a group of crustaceans that has been around since the Cambrian. There's only a handful of species left, and on coral reefs per se, they live in the coral matrix. So there's just some really amazing animals. And many of the things that live in the reef matrix are basically cave-adapted organisms because they live in these dark little crevices, and they have small eyes and long appendages because they don't rely so much on sight. They live in a dark environment once you move deeper into the reef matrix system. Other bizarre things you come across here. This is a clam, okay? You can see the shell. The shell is right here, that little white outline, okay? It's internalized, and that's all that's left of it. Not what you think of with most clams. Here is another clam. This one has the two shell valves flattened out, and it crawls around on it and forms all sorts of bubbles on the edge of its mantle. Just really bizarre organisms. And the story goes on. I mean, I could show you slides for quite a while. And there's just an incredible range of critters that make the reef their home. David Lichwager, who's a National Geographic photographer, worked with us on one of our surveys, and his thing has become, in recent years, to look at a cubic foot of space in some living ecosystem. So he took a cubic foot, you can see the fish and the cubic foot around the area, on a reef in Maury in Polynesia, and after watching fish go through that area and documenting the sort of the mobile roving organisms that go through there, he physically removed that cubic foot of reef and took it apart in the lab with our help, and this is what came out of it, okay? This is something like 400 individuals that were in that single cubic foot of coral reef. This is what the coral reef is about. This is what you don't see when you don't look inside it. So why live in the matrix? Why go in there? What's the point? Why not just live out in the outer part of the reef and enjoy the scenery and see all the beauty that's there? Well, same story as everywhere on the planet. There's things that will eat you. There are lions and tigers and sharks. Oh my. This is what happens to a sea urchin if it happens to be broken. On the left side, you see a feeding frenzy of a broken sea urchin that when I moved the rock, it broke open, and this is the fish community that congregated on it to feed on it, okay? On the lower right is a helmet shell that dealt with one of those incredibly spiny, the black sea urchins that can really get you, and this helmet shell just went right down on it and chowed it off. So even if you're that protected, you can succumb to predators. Otherwise, you get the grazers. If you're a sessile organism on the surface of the reef, there's these marauding brigades of grazers that will feed on you. And of course, this is why reefs are what they are, because these guys keep the algae down. And things with defenses, things with skeletons like corals that sting, can survive this onslaught at times, but not always. Here's a Porites colony that got chewed by parrotfish. Okay, so even there, these grazers can get into you and chew you down. So how do you survive in an ecosystem like this? I mean, most ecosystems have a lot of biological interactions, but the more diverse the system is, the more severe these become. The escalation of predator-prey interactions, competition, becomes pretty intense. And there are a number of strategies, and the most obvious one, which is the story of this talk really, is you move inside the reef. You move into this reef matrix. But there are also structural defenses, there's chemistry, there's camouflage, and you can also live on other organisms and hide on them and take advantage of them defending you. So here's a little crab on a soft coral looking out, and Kathy showed an earlier picture of a snail that had polyps on its back. This guy has polyps on its back too, and it's not polyps, it's itself looking like polyps. Structural defenses are usually skeleton, right? So the corals themselves have it, coral and algae have it. On the upper right is a protist, it's a foraminiferan, which also has symbiotic algae in it, little green algae. On the bottom is a sea star, Coolceta, which has a pretty well-armored skeleton body that again protects it from predators. You can be toxic, and many toxic things are colorful to advertise the fact that they're not good to eat, so that things don't bother them. Sponges are excellent chemists. Well, they are not actually. They have a lot of bacteria inside them that are excellent chemists, and they make some phenomenal secondary metabolites, which are really good for potential cures for all sorts of human diseases. So biochemists and medical researchers study the chemistry of sponges for this reason. And then if you feed on sponges, like that sea slug in the upper left, you become toxic if you pick up that chemistry and can hold it in your body to ward off predators. The flatworm in the upper right does the same thing with ascidians. And then this is an endemic sea urchin in the Red Sea, the red one. And every spine tip, you can see those little bubbly things on the spine tip. Those are venom-tipped spines. You touch one of those things, it's like you got a really bad bee sting. You can also do camouflage, and a lot of things live on corals and hide there. And you can see the clingfish in the upper left, a porcelain crab in the upper right, and an eggshell cowrie below. Or you can live in somebody else. Almost any large organism has large symbionts living in it, and of course, everybody has little tiny symbionts living in it. That little shrimp there lived inside this scallop. The shrimp itself can have isopods living in it. So you see that bulge on the carapace of the shrimp above? If you open that bulge, that's what pops out. The big one is a female, and the little one is the male. The female is this huge egg-making machine. It's just producing eggs and releasing larvae constantly, and there is her pocket husband, keeping her company. Barnacles. Everybody has barnacles. The upper slide shows you what happens to the lobster when it doesn't brush its teeth. Those are lobster mouth parts, and every one of those little white things is a barnacle growing on it. There is a black coral with barnacles on it, and you can see them mimicking the black coral to such an extent that they make these barnacle polyps on their body to hide from predators. But barnacles are in everything. Corals, for example, have a huge number of barnacles that live in their skeletons. Here's a sea cucumber. You see the snail on its mouth? It's a vampire snail. It sucks its blood. It inserts its proboscis into the blood vascular system of that sea cucumber and feeds on its blood. These echinovampires are probably the most diverse group of snails on reefs. There's hundreds of species in this family, the Eolimidae, and every single one of them is a parasite of echinoderms. Some of them stay on echinoderms all life. Some of them go on and off the echinoderm to feed, sea stars, sea cucumbers, sea urchins. Some of them form galls. In the upper right, you see a sea star where we dissected the snail out of the arm of the star. It was embedded as a gall inside that arm. Copepods. Well, some of the copepods look like little crustaceans, but not all of them. Some of them are so modified because of their parasitic existence that you can't even recognize them as arthropods. Daisuke Ueno, who's also spent time at KAUST working with us on these biodiversity surveys, has made this picture of just some of the animals he studies that are these really highly degenerate, modified copepods. Among other gastropods, also parasitic, are this family of snails, all of which basically suck sponges. They go onto sponges and they insert their proboscis into the sponge and suck on its contents. And it goes on and on and on. The most diverse family of clams in coral reefs or in the world is this one. It's called the Galeomatidae, and they are commensals. And they have these degenerate shells. I showed you a couple before. And these colorful bodies and tentacles. Some of them develop eyes. They crawl around. they just about regrow their head. One of the really weird things about clams is they are mollusks that lost their heads, and then here is one group of clams that's basically trying to regrow that. So why be on the reef surface at all? Why would you want to be out there on the reef if everybody's trying to eat you? Well, to feed, right? The inside of the reef gets its food from the outside, and much of the food on the reef comes from the sun. The reef basically is a garden of animals and some algae and plants. Phototrophs like the algae, or photosymbionts, which are animals that have algae in their tissue, and as you've heard in the other talks, recycle the animal waste and the sugar that, and fats and so forth that are made by the algae between the two partners in that symbiosis. And then if you are an animal that doesn't have photosymbionts, the most common way of living, at least initially, is to suspension feed. And if you suspension feed, you're sucking on plankton from the water over you, so you also need to be in the reef. Photossymbiosis is incredibly widespread on the reef. Most people think that that's something that corals do and it ends there. It doesn't. There are photosymbiotic clams, the giant clams. A lot of people know about that. The bottom one is an ascidian. It's a sea squirt, a colonial sea squirt with algae. That's a sponge on the right. There are flatworms. There are forams. There's a whole diversity of things that are photosymbiotic. And not only are the animals diverse in these associations, but so are the algae. Some of the algae are dinoflagellates in corals. Others are modified diatoms and several other major groups of algae that partake in these symbiosis. Once you live on the reef surface, of course, if you're sessile, there is a whole war, turf war that goes on, because the primary thing that sessile organisms compete for in a limited system like a reef surface is space. And what you see here is two corals, a Porites on the left and a Goniopora on the right, duking it out. They can be very aggressive and try to overgrow each other. The Goniopora can grow these really large polyps as aggressive interacting polyps that are defensive and try to whack their neighbors. And seawater just, the currents just take those polyps and move them back and forth. And when they land on the Porites, the stinging cells from that polyp just burn holes in the other coral. And in this case, you know who's going to win out in this interaction and who's going to take over the space that's there. And this happens in everything. On the upper left is a coral being overgrown by that black tar. That black tar is a sponge. It's one of the most aggressive space occupiers on reefs where it exists. The black color comes from symbiotic algae, from symbiotic bacteria, actually, blue-green algae, cyanobacteria that live in it. On the upper right is a fire coral overgrowing a stony coral. And on the bottom, of course, is what we are often worried about in coral reefs, when the algae get an upper hand, in this case, cyanobacteria, and they overgrow the reef-forming organisms like corals. Now, the filter feeders are usually soft-bodied animals that need to protect themselves, that get in trouble if they don't. And here's an example of what happens. The big picture shows you a fire coral colony with those round holes in it. They are worm snails, and then little things sticking out of them, which are segmented worms. And they all create tubes that grow enmeshed with the coral and gain protection not only from their own skeleton, but also from the stinginess of the fire coral itself. Here are worm snails. Those long things are actually snails, and their shells become uncoiled, embed in the coral. And then see this? These are mucus nets. They put snot out all over the reef, and that snot captures particles that land there, plankton and so forth, and they draw it back into their body and eat it. And then, of course, people like to eat these. In Polynesia, these are a delicacy. You'll see people on a lot of shallow reefs going out there with hammers and just hammering these out of the reef systems for dinner. And they are quite tasty. And that's why they do what they do. Christmas tree worms, another group that really embeds itself in reefs. Those are scallops, really colorful scallops that have got the ability to nestle into coral and bore into coral. And it goes on and on and on. And one of the basic things that life does on a reef is escape into the coral. So if you're a suspension feeder and you need access to the water on top, you're going to try to bury into the coral. One of the most severe ones out of these are sponges. That little pinkish-orange spot there is the exhalent opening of a sponge that extends deep into the coral surface and excavates it. The sponge body itself is underneath the coral, eating out the inside of the skeleton, leaving the surface intact except for that snorkel. Here is another example of that, and you can see both the incurrent and the excurrent snorkels on that sponge, and they totally excavate the inside of that coral colony. Then you have snapping shrimp that excavate. So here is an opened gallery that shows the players. They're always in a husband and wife team, right? And then they tunnel underneath the coral and open up little holes to reach out and feed from. And they come in a couple of flavors. Probably a lot of you who dive seen these openings on the reef, these galleries that are full of algae. They actually farm algae in it. And the animals that make it are those snapping shrimp on the upper right. There are crabs that excavate into living corals. They're called gall crabs. And they make holes into the coral, and we don't even know what they do, although they tend to have gardens around them. So I bet you they are feeding not only on coral mucus, but also on algae potentially that grow in those gardens. And here are a couple of examples of them taken out of their home. They look like little ticks because they're parasitic and they can grow big things, big bodies like that. Date mussels are related to the eating mussels, same group of bivalves, that bore into the interior of both corals and just reef rock and excavate large galleries in it. So what happens on the reef is that a lot of organisms hide in the reef. The consequence of that is erosion. We call it bioerosion when it's done by animals. It can be mechanical. It can be chemical by acid. The consequence is that you end up with a lot of holes in that reef matrix. And there's also chemical erosion and both biological and chemical deposition of carbonate. So the interior of the reef is this Swiss cheese thing that's completely being reworked all the time, and the space is enlarged and contracted and so forth. What you're seeing on the upper left is the snorkel opening of the bivalve that I exposed by breaking that piece of reef in half, okay? So the opening, the snorkel is up on the top, and then there's a long path leading down to the clam that was inside that reef. Now, when you live in the matrix, sometimes you might want to come out. Some animals come out of the reef at night to feed. Because if you're completely inside the reef, you don't get as much food as if you would emerge. At nighttime, there are less predators, so it may be a safe time to come out. Many animals just extend out of the reef at night. If you go night diving, you'll see a lot of tentacles, like these ophiuroid arms, these brittle star arms extending out of the reef to feed with. A lot of the filter-feeding crustaceans, like porcelain crabs and squat lobsters on the right, come out to the tips of the coral branches in which they hide during the daytime and use their legs to gather plankton and feed on it. It's completely a nighttime thing. Other things just come out to feed. Many hermit crabs you wouldn't see in the daytime. Some areas you see some, but in the real diverse parts of the reefs, hermit crabs are very early out, but they come out and they row around at night looking for food. Snails come out that wouldn't be out in the daytime and look around, and of course you get all this other diversity that comes along with it. That sea anemone is always on hermit crabs. It's carried around by hermit crabs, and it's a symbiotic system that emerges because the hermit crabs do. The other reason you come out at night is for reproduction. A lot of animals, basically, if you want to have the eggs dispersed in the seawater, you need to come out of the reef to do that. And one of the most spectacular examples of these are segmented worms. Palolo worms are one example of that basically subdivide their body and send their tails out to go reproduce while the rest of their body stays in the reef. And this is synchronized incredibly, so there are certain nights where they're just out there in huge numbers, usually the same nights that corals spawn on, and this is what it looks like. In some parts of the world, in Samoa and Fiji, they eat them. So they're collected in large quantities, and you can have Palolo worm omelets, quite a delicacy. And here is a Palolo worm growing. The tail, the orange part is full of eggs, and that's where it's going to break off and go up as a swarmer into the seawater and reproduce. Here's another one. It's a different polychaete worm, but this is, again, a reproductive swarmer. And you can see that when they go into reproduction, they grow huge appendages so they can swim in the water column effectively. They have really large tentacles to sense the environment around them. They completely change their morphology because now they are reproductive. And here are a couple of other pictures of reproductive worms that are out in the water at night that live otherwise inside the reef during the rest of their existence. And then even some sea urchins do. This is a strawberry urchin. Very, very, very few people have seen it, because the only way you can see a strawberry urchin is if it comes out to spawn or if you find it well inside the reef. Okay? It's a spectacular sea urchin with that lives only in the interior of the reefs, except during reproductive bouts when it comes to the surface. So how do we study this? So I basically have specialized on documenting biodiversity by large-scale surveys, because this is the effective way of doing it. And I apologize, we changed the title of the talks. It was going to be Mare Incognitum, which is one talk, and then, anyways, it became the Inside Story talk, but but it's really both. So here we'll talk a little bit about how you can learn about the world. And what we do is get a bunch of people together. Here on the left is one of the Cows cruises off on one of the reefs, and you get a lot of people into the water to bring up specimens into the boat, and there you look at them. So you do a couple of very different style of collections. One is specialist collecting, when somebody who really knows a group collects. The other is mass sampling, when you bring stuff back to the boat to take apart, and then you triple document it, and I'll tell you about it all. And this is sort of where we've been so far. So as Mike mentioned, we work all over the world to document marine life, and because the Indo-Pacific is so connected, we get to know a lot about this fauna through these surveys, and a lot of these surveys will recollect the same species and tell us a lot about connectivity patterns as well. So I'll use examples from three areas where we've done a lot of work. One of them is Guam, where, as Mike mentioned, I spent 9 years on the faculty, and we did a lot of work there with both the local staff, students and amateurs in the area, and resource surveys that we did in the colored areas. I'll also talk about Moria, which is in French Polynesia, where we had a project to document the entire ecosystem and do DNA barcoding on every species in it. And then, of course, I've been working in this area since about, in Saudi Arabia since about 2012, and in Oman since '99, on and off, and a little bit in Djibouti to document the fauna of the Arabian Peninsula. So I'll use examples from those three places. So how do you collect stuff? What do we do when we get in the water? Well, some things you do, you bring it around people who really know the animals and know where to look for them. So this is Terry Gosliner, who I call the Slug King. He's a specialist on sea slugs. And you throw Terry in the water, and I'll go in the water with him, and I'm a pretty good collector, and I'll come up with 5 sea slugs, and I'll give it to Terry, and he looks at them, it's like, common, don't want it, don't want it, don't, yeah. And he has a little jar, and it has 40 species of sea slugs in it, right? And I didn't see any of those. But he does, because he knows where to look, he knows what they look like, and he notices them. Okay, so people like him can find stuff that the rest of us rarely encounter. And then, of course, he writes big papers and studies and so forth about that. All of us do specialized collecting that work in the field. And one of the things you want to know is the microhabitats, the kind of habitats these organisms live in, under rocks, inside coral colonies, in commensal associations where there's organisms and so forth. You need to learn the biology of the animals to find them. You also use funky tools that really target certain organisms. This is a yabby pump, which is just a big slurp gun. And you can use it to pull out yabbies, which are ghost shrimp and mud shrimp from their burrows. We want to photo document. We don't want to photograph what we collect. And a lot of the things that are attached to the reef are best photographed in situ. So typically, people will dive with multiple cameras. This is Daisuke Ueno, who works on Copepods, and he always dives with 2 cameras. The other way to collect is to mass sample. And this is a plankton tour, and it gives you an idea what a mass sample is about. You do a plankton tour, and there's going to be hundreds of species, thousands of individuals, and you pick through it and document what's in there. Another way to mass sample is to vacuum the reef. This is an old vacuum system where you're using a scuba tank to drive air through that long pipe and suck water out through it into a mesh bag, and then you collect what's in the mesh bag to get your critters. And here is a more modern system developed by Twilight Zone divers that couldn't use tanks because they would run out of air too fast. This is driven by an ROV motor on a battery, and then you just take it down underwater with that bucket and suck up all the little things that make up most of the reef. Okay, so for mass sampling, you bring back other things. If you want to look at the cryptofauna, the least destructive and most effective way to do it is to bring back rubble. Okay, dead pieces of the reef from the bottom, dead coral heads and the like. You take a bucket worth, and that will set you up for the rest of the day. You go up onto the top of wherever you work, in this case a boat. This is on one of the cow surveys, and you use a hammer and chisel to take apart that dead coral and look inside it to see what all is in there. And you pick and you pick and you pick, and then you sort the animals that you get, and this is the sort of stuff that comes out of the matrix when you do that. And a lot of diversity. Here's a nice video we did in Moria that shows some of this. And this shows using arms, which are these autonomous reef monitoring structures that are stacked settling plates. And these mimic the reef. So you don't even have to take the reef apart. You put one of these down, the animals move inside it, then you pick it off the reef a year or two later, and you have the cryptobiota at your fingertips. So these guys put a cover over the arms unit that has Nitex meshing on it to keep anything from escaping. Then they bring it into the boat. You can see how much life has grown on this structure since it's been there. The critters that move into it are pretty similar to what's on the reef. They're the same species and a fairly similar composition. You take the arms unit out, and it disassembles. These plates come apart, and look at how many things are growing, accessible things on it. Those red and orange things are sponges. These white and red things are tunicates, or sea squirts. You get bryozoans. You get all sorts of life. This is not what you think of as a reef looking like, but that's what the inside of the reef is like. This is the very, very typical cryptobiota community from the inside of the reef. And that's the exposed part with coralline algae. Then you take all of the water that was in there and you filter out the organisms and go through them and pick out what was in it. So this is all from a single arms unit. These guys are, there's a major arms project being done by the Red Sea Center here in the Red Sea, and they're getting the same sort of things out of that. Each of the cups is a different organism. That's one arms unit, okay? And then if you do a large biodiversity survey, you get a lot of people together, microscopes, etc. And what you do is you sort these animals you collected into individual species, you do your best identification on them, photograph them, take a tissue sample, voucher them, and then you take them back to the museum. Once it's back into the museum, you arrange them, you database them, you might do DNA sequencing on them, and then you send them out, if you don't know them already, to experts. As Kathy mentioned, if you work on soft corals, there is 1 of 5 people in the world you can send it to get an ID on it. So you send it to that person, and hopefully they'll look at it and help you identify it. Much of what you find in these surveys is new. For example, squat lobsters are phenomenally diverse, and now there is finally somebody in the world who's taking them seriously, a guy called Enrique McPherson, who's in Spain, and he's been churning out paper after paper after paper describing these animals. In one of the recent papers, he described 92 new species in the genus Galathea, out of 144 total known. In our Moria survey, 8 of the 9 Galatheas we picked up were undescribed, were new species that were described in that paper. So once you do a survey like this, you just end up with the real diversity that lives on the reef. This is the results we published from Guam after I left the island, about 5,600 species there. And what was interesting is they did a big publication in 1981, and the yellow lines represent how much more we documented in the following 20 years, okay? Even in fishes, which is the last graph on the right, you can see a huge increase in diversity in just 20 years from people studying the area more fully. Now, I want to say a little bit about what we've been doing here in the Red Sea and some of the results we got. So since 2012, my lab and I have participated in 6 cruises that KAUST has organized, with about typically 1 to 4 people specializing on invertebrates per cruise. We got about 7,000 specimens from these, about 30,000 photographs, and about 2,000 species. And then we have also a lot of material from Djibouti and from Oman. And you find a lot of cool animals. So I'll just share a couple of the cooler things with you. One of my favorites that I wasn't here to see, but Daisuke was here in 2014, and he collected it, is a thing called Lobatolamphia tetragona. This is a ctenophore. It's a comb jelly. This is a group of animals that usually live in the water column. They're pelagic. They swim around. They're really weird. Some people think that these are the first branch of the entire animal kingdom, even below sponges. And if that's true, then nervous systems evolved twice in animals. And that's a very intriguing result. Anyways, this is an entirely pelagic group, except for 2 branches. The top branch there called Platectinidae, which are benthic, which crawl on the bottom, and this thing, Lobatolamphia, that independently evolved a benthic lifestyle that was described from Japan and until very recently was only known from Japan. And we found it here in the Red Sea. And it's just this totally bizarre benthic ctenophore, an animal that's an entirely pelagic phylum that has a benthic member. Those little pearly things you see in there are babies that it broods. Sea cucumbers are what I focus on when I sort of play a specialist in taxonomy. And so, of course, we've been looking at those in the Red Sea. There is a total of 63 species recorded in the literature. We got about 42 of them. 14 were new records, and 16 were new species. 38% of those species that we have sufficient information on are endemic to the Red Sea. And that's based on both morphological and genetic information. So you can then look at patterns in the Red Sea of why are they endemic. So the problem with the Red Sea is it's a very young basin. Okay, it's a couple of million years old. That's not that young. But when the ice ages came, the basin got isolated and the salinity increased tremendously. And if you look at the planktonic fossil record, it shows that most planktonic species went extinct in the Red Sea during the ice ages because it was so salty. And some people say that it was completely defaunated. All of the animal life died in the Red Sea. That clearly must not be the case because there's a lot of endemism. But it sets up this paradox of how come you have these endemic species in a basin if everything died in it 15,000 years ago? And this just shows, of course, the depth contours in the basin, that the mouth of the basin is only 137 meters deep, and the sea level fell by about 120, 125 meters in the ice ages. So there are three potential explanations for this paradox. One of them is the endemics are very young, that they evolved in the last 15,000 years. One of them is the endemics survived in refugia, somewhere in the Red Sea that didn't get so salty, or they managed to cope with the salinity, which we know from experimental tests a lot of these things can do, most of these things can do. And the other is that they actually are not truly endemic to the Red Sea. They spill out and are outside of it, say, in the Gulf of Aden. So with a data set like sea cucumbers, you can look at that. And out of the 19 endemic species that we collected, 8 are found not only in the Red Sea, but we also found them in our surveys in Djibouti and Oman. 9 of them were not found outside there, so many or most exist outside the Red Sea basin. Oops, this is a duplicate slide. Sorry, that wasn't the slide I wanted to show. This was the slide I wanted to show. 15 endemics collected, 15 endemics that we collected. The rest of it was all the literature things. 15 endemics that we collected in Red Sea, 12 were collected by us. 9 of those are recorded in the literature from Djibouti and Oman, and only 13 endemics were not collected outside the Red Sea. 9 of those are only known from the first time they've ever been picked up, and they've never been collected anywhere else, even in the Red Sea. And 2 of those 13 that are that we did not collect in the Red Sea were collected by others outside the basin. So most likely, most of these animals actually are not truly Red Sea endemics, right? And here is an example of one. This is genetic data on this species, Holothuria cinerescens. So if you look at the tree in the upper right, the first branch is a specimen from Oman and Red Sea, and those are indicated by red on the map, and the green dots are samples we had from our other surveys of this species that show a second branch in that. So this is a species that recently diverged into a population in Arabia and a population in the rest of the Indo-Pacific, and the Arabian population is not a Red Sea endemic. If you look at all of the species that are restricted to the Red Sea area, and you look at their age using molecular clocks, what you see is an incredible spread in ages, but the ages are old. These are in millions of years on the y-axis. So the age of the endemics ranges from about 500,000 to 5 million years old. So they're strung out over time. They formed gradually over the last 5 million years or so, and they're not young, and they weren't formed at a single time period. There's one exception. This is one of my favorite animals. When we first collected this in Djibouti and then again in the Red Sea, I was sure we had a new species, and it is very distinctive. Its closest relative is a species you see in the upper right, which is known from the rest of the Indo-Pacific, but genetically they are basically identical. So this is either a very, very young species, or there has been gene flow since its form between the two populations that mask the divergence. So what we can conclude when we study a group like that is that young endemics may happen. We have one potential endemics, one potential example. Endemics survive in Red Sea refugia, maybe some. We don't have enough information. Nothing really convincing that's truly endemic to the Red Sea in sea cucumbers, but there are examples in other organisms. And Francesca just showed you a coral that is like that. And of course, there are some fish. Some of these live up in the northern end of the Red Sea, like in the Gulf of Aqaba, which is also known to be an area that have received more fresh water during glacial times, thus could have had a more reasonable salinity condition than the rest of the Red Sea. And of course, our main results have been that most of the endemics that were thought to be Red Sea endemics also spill out into at least the Gulf of Aden. So what do we learn from large-scale biodiversity surveys? Well, what you see when you do one of these surveys how incredibly effective they are. A single survey can document literally thousands of species if you have the people necessary for it. Technology really speeds up how fast you can document things and how fast you can determine what they are, and this includes mass sampling, it includes digital photography, and using DNA to help identify species. How many species are there? Well, in a typical reef system, easily 10,000 species. I'm sure there's more than that in the two-wall reefs just off here. How does diversity vary among groups? Most species live inside the matrix of the reef. They are what we call cryptobiota, and or they are symbiotic on other organisms. And how much diversity remains undescribed? Most of it. And what's really a reality for taxonomists, and this is what we're dealing with today, is that the diversity of species is highest in these tropical places, but the study is concentrated in Europe and North America. So the top graph shows you species density of fishes, sort of what we know about fishes, which are the best known marine group, where the diversity of fishes are. And the bottom graph shows you how many records there are of marine organisms in the largest biogeographic database out there, OBIS, and the hotspots are in Europe and Eastern North America, which are some of the lowest diversity areas. So the tropics are a huge job that remain to be done for documentation. Really the thing that drives me and I feel is one of the most compelling reasons for these surveys is that we have to find out what lives on these reefs today if we're going to have the information to manage them as best as we can for tomorrow. And the knowledge that we gain today are what all of future generations will have available to them often, at least about what healthy ecosystems look like and what many of the species look like. And Douglas Adams, who some of you might know as the author of The Hitchhiker's Guide to the Galaxy, has a really compelling and sort of bittersweet book about it called Last Chance to See. And with that, I'd just like to thank all the people that I work with. These are all very large projects. We have incredibly capable crews back home at the museum who process material and host visitors and send loans out in the field, both just divers and taxonomists that come along here at the Red Sea and elsewhere. And then a lot of the work that we do involves sequencing and phylogenetics. So a lot of people involved in that, and of course funding agencies. And thank you.
Thank you. We have time for a couple of questions. There's one microphone coming.
How does this work? Oh, wow. Thank you so much for this talk. I'm pretty sure I'm not the only person in this room who's blown away by the images and the diversity that we as students are apparently still too far away to realize what's even happening on this planet. It's great. So I'm wondering, you're basically taking out all the things you can in order to categorize them and put them into literature, or try and find out what's out there. But have you ever encountered, or have you ever found a species where you thought that might be one of the last ones? Or have you ever taken out species to an extent that it would have affected the environment? Do you know what I'm getting at? Because
Sure.
Obviously you're taking it out and you need to kill it.
So
right?
I lived on Guam for 9 years, and Guam lies in the West Pacific typhoon corridor. It gets hit by one typhoon on average every 2 years. I've seen about 10 major typhoons hit Guam while I lived there. One typhoon takes out more reef than every scientist on this planet has ever collected in their lifetime, and for generations back to Linnaeus. So that's the perspective. I mean, I'm very much a conservationist. I'm working entirely toward conservation, but without getting your hands on these animals, you can't study it. All of the coral collections, all of the museums of the world amount to roughly an acre or two of reef, okay? So think of it in that perspective. We bring a bucket of rubble up to the boat to look through and look at the size of the reef down there and look at the next bleaching event or typhoon or crown of thorns event that comes through there, and then think about it in that perspective. And I think that explains why we are willing to do the destruction that we do. You cannot study something without getting your hands on it. It just doesn't work.
Have you ever found a species that you think might not exist anymore?
So I'll tell you one story. So in invertebrates, they're so small, we haven't a clue. I mean, we're always finding new things, so how would you ever know you found the last thing, right? Yeah. I mean, most of them are, if they're rare because we are not where the species at. So there are very few extinctions documented yet, partly because it's hard to do, partly because we are not as far along yet in what climate change will bring. It's coming. But one interesting example actually is in Oman, and it's a cowrie, Cypria tuleri, that was only known from Oman from a single habitat. It's actually a living fossil in the cowrie lineage. And it has direct development, which means it doesn't have planktonic larvae, okay? So it cannot disperse very far. And there was only one population known from about a mile of shoreline. And shell collectors found out about it, and they paid locals to collect it, and they went there to collect it. And when I was in Oman in '99, we were working on cowries, and I went to the place, and I said, I'm going to get some to sequence, get the DNA out of it. We couldn't find it. We found out a whole bunch of shell collectors said population is gone. That was the only living population known of this species. So there is an example where it was collected to extinction, except it was found again in a different population later on. So thank God. But there I was convinced that that species could have been collected to death by collection, which is otherwise I have never, ever heard of an example like that.
Great, thank you so much.
I have a question. So when you're finding all these new species in sediments, and I guess wherever you collect them, how do you know that they're native to the area?
You don't.
You don't.
So yeah, that's a very good question. So how do you tell that? So you place it in context. So now with genetics, we have a lot of tools to really look at connectivity among populations and among species. If it's new, you really don't accept in context. So it belongs to a group that's native to that area, that's not known from far away, you're pretty sure. If you're in a pristine reef, or not pristine reef, if you're away from ports and estuaries, 99.99% of what you find is native. We very rarely found marine invasives out in reef systems. There are very few examples of that. They do occur. Most of them occur in ports, in estuaries, in enclosed lagoons where there's a lot of shipping traffic and they don't span very far. So just based on that as well, we don't think they're that common. They can happen. And then once you think there is something that could be an invasive, collecting populations from elsewhere is necessary really to put it into that context. And until you have that, you cannot be sure. Sometimes, though, you know that it belongs to a Caribbean group, it has to be an invasive, and mostly in port situations, and then you can try to go after that.
Thank you.
Any other questions?
Okay, I'm off. Okay. Thank you.